Hello, here we are back for video number 2 on the fluid balance chapter. So in the first part of this chapter, we talked about where we hold fluid in our body, intracellular versus extracellular compartments. We talked about which electrolytes are found in each. So we know that potassium is found in higher amounts in the intracellular compartment, and we said that sodium is found in larger quantities in the extracellular compartment. And lastly, we talked about the fact that you should know what percentage of body fluid is held in each. So about 2/3 of our body fluid, 65% or so, is housed in the intracellular fluid, whereas the remaining 35% is housed in the extracellular fluid. Then we moved on to talk about water balance, what hormones are responsible for that. And then we talked about electrolytes, what they are. And then we talked about potassium and sodium and how their homeostasis is regulated. And lastly, we touched upon calcium. So this leads us to the last portion of this chapter, and it's a doozy: acid-base balance. So in addition to being able to maintain water balance and electrolyte balance, of course, if not even more important, is our acid-base balance, because we know that our bloodstream operates in a very narrow range of pH. And if we go outside of that range, 7.35 to 7.45, very serious things can happen. So off we go. So just some things to review before we start getting into the nuts and bolts. We already know that our normal blood pH range is 7.35 to 7.45. We know that the blood is considered to be extracellular fluid. We know that there are things that can influence acid and base balance happening all the time. So you see, this is what I'm talking about. Even though you may not realize it, almost all the time throughout the day, there's things that can enter the bloodstream that can threaten to move the pH out of that range. So it's not just when you think of the obvious thing. In other words, it's not just when someone is in a diabetic ketoacidosis situation that we worry about acid-base balance. There's everyday things. If you haven't eaten for a while and you start to burn more fats, we're going to have more ketones. So there's threats to altering pH all the time. And what that means is our body has to work 24/7 to maintain acid-base balance. And because of the systems we have in place, the body is typically very good at maintaining that pH range. Okay, so our main discussion in this chapter as it relates to acid-base balance is going to be on buffer systems. And that's what the lab is that goes along with this, the buffer systems lab. Before we talk about that, though, we have to talk about some definitions. Some of this will be a review. Some of it might be new to you. We are going to keep things pretty general, so if you have a chemistry background, please fight the urge to go into more detail in terms of this discussion. We're going to keep it pretty superficial. Make sure, first, that you can define acid and base. We did this in A&P 1 back in the chemistry chapter. So just review. When we talk about an acid is anything that releases hydrogen ions when put into a solution. Releases hydrogen when put into a solution. I always give the example of lemon. You know, lemon juice is very acidic. So if I have a glass of water and I squeeze lemon juice into it, what's happening chemically is that lemon juice, the lemon juice droplets that are going into the water, once those lemon juice droplets get into the water, they are chemically releasing hydrogen into that water solution. That's what an acid is. Anything that when put into a solution releases hydrogen. A base would be the opposite. A base is anything that takes up hydrogen ions when put into a solution. Baking soda, something like that. If I drop baking soda into a glass of water, even though you can't see it, what's happening chemically is if I drop baking soda into that water, the baking soda is taking hydrogen from the water and absorbing it. So it's picking up hydrogen ions. It's true that bases also release hydroxyl, but you do not have to know that for our discussion. Before we go on, I just want to point out that if we look at solutions, any solution that has a high hydrogen concentration is said to be acidic. So I just want to make sure that we're clear on the relationship between amount of hydrogen ions and acid versus base. Because an acid releases hydrogen, that solution becomes acidic. So anytime a solution has a lot of hydrogen ions, it is acidic. And we know that acidity is represented on the lower end of the pH scale, right? What that means is the opposite is true for base. Anything that has low hydrogen ion concentrations is alkaline or basic. And, of course, basic or alkaline is represented on the higher end of the pH scale. So just review that. All right, now let's talk about strong versus weak acids and bases, because this is what's going to matter for our chemical buffer systems. Let's first talk about strong. A strong acid would be something like, oh, why did I put yellow? Why for yellow? What I meant to put is lemon juice. I was thinking it was yellow. Haha. Lemon juice. Or if you want to think about an example in the body, hydrochloric acid. We talked about that in the digestive system. Very acidic. These are examples of strong acids and bases. And these are examples of strong acids, Strong acids will release their hydrogen and dissociate completely. So if we have a strong acid, and what we mean when we say dissociates completely, imagine, as I said, if we drop lemon juice into a glass of water, we know that those lemon juice droplets are going to release hydrogen. If it dissociates completely, that means that the hydrogen ions are released and spread out into the solution completely. Let me say that again. So if it's a strong acid, and we know an acid releases hydrogen, it means that those hydrogen ions are released and spread out completely throughout the solution. So if we have a strong acid that dissociates completely, it means that the hydrogen ions spread out in that solution completely. So whatever size that complete, how about I can't spell? Completely. I think I spelled that wrong, but apparently I can't think right now, aside from acids and bases. So if we have, when we say dissociate for an acid, those hydrogen ions spread out completely into the solution. A base would be likewise, but instead of spreading out completely, a strong base picks up hydrogen ions from the whole solution. So a strong base picks up hydrogen ions completely from the whole solution. Because the opposite of that is a weak acid or base. And a weak acid or base does not dissociate completely. So if we compare, let me change a different color here. So we said, let's use green. So we said a strong acid was like lemon juice or hydrochloric acid. And in a strong acid, the hydrogen ions spread out completely into the solution. Let's compare that. Let me use a different color. Let's compare that to a weak acid. In a weak acid would be like carbonic acid, which we're going to talk about in a little bit. In a weak acid, hydrogen only spreads out a little. This is my own definition, not so much scientific, but bear with me. So in a weak acid, the hydrogen ions do not dissociate completely. So what that means is, instead of hydrogen ions spreading out into the solution completely and evenly, in a weak acid, the hydrogen ions just spread out a little. Or if it was a weak base, like bicarbonate, if it was a weak base, the hydrogen ions are picked up just a little bit from a small area. Versus in a strong base, the hydrogen ions are picked up from the whole solution. All right. In our chemical buffer systems, we're going to be utilizing weak acids and bases, and we'll talk about why later. I know you can't wait. Okay. Just to go back a little bit, I want to touch on a little bit about, I want to touch upon a little bit about a base. So let's go back to blue, because I like blue with this color background in the PowerPoint. It makes me feel all happy inside. When we talked about a base, we know that a base picks up hydrogen ions. What I want to point out is, oftentimes, when a base is released into a solution, and when that base in a solution begins to pick up hydrogen ions, a lot of times that base picks up hydrogen in order to form water. And that's what's going to happen in our chemical buffer systems that we'll see in a minute. Please try not to overthink this. If we can just imagine, I gave you the, let me just go back. If we just imagine we drop something that is basic or alkaline into water. And what are those droplets of the basic solution going to do? They're going to pick up hydrogen from the solution. A lot of times, in the process of picking up hydrogen, that base goes on to form more water. Because what is part of water? Water is H2O. So when a base picks up hydrogen from the solution, one thing that it can do with that hydrogen that it picked up from the solution is form water. And we're going to see that come into play, but keep it simple if you can as we go through this. We already know that a base, when put into a solution, picks up hydrogen ions. And now we're just saying one thing that the base can do with the hydrogen ions that it just picked up is it can form water. Keep it simple. Okay. We talked about strong versus weak. A strong acid or base will dissociate where the hydrogen spreads out completely. In a weak acid, it only spreads out a little bit. You do not have to know these equations. Moving on. Here's where we get to the real fun part. Sometimes I sing to myself at home. And I guess I do so even when I'm recording a lecture, because I get so excited. Okay, so in the body, we have these buffer systems. Okay, let's talk about our two buffer systems. We have our chemical buffer systems. That's one type of buffer system we have in our body. And we have our physiological buffer systems, which is another type of buffer system we have. We use the chemical buffers as a short-term means of buffering against pH changes. Short-term. So chemical buffer systems are our short-term way to maintain blood pH or to buffer against large pH changes. So the chemical buffers kind of act as a band-aid in the short term. It's not permanent, but in the short term, it can buffer against changing the blood pH. But we're always going to rely on our physiological buffer systems to do the long-term pH buffering. Oh, there's my cat again. She always knows when to show up. It's like she's a little ham and she knows. Okay. So we have two types of buffer systems in our body: chemical buffers, which we use in the short term, and then physiological buffers, which we will utilize in the long term. And these physiological buffers are more permanent. Let me add that. These are permanent ways that we can, for example, get rid of hydrogen ions. And then just review what a buffer system is. So both of these are a way that our body can buffer against large pH changes. So we have that range in our bloodstream, 7.35 to 7.45. So we need to have these buffer systems so that when the pH in the blood starts to go down, we can enact these buffer systems to make sure that it doesn't drop below 7.35. Okay, let's talk about the chemical buffer systems first. So the next several slides are going to be addressing chemical buffers, which are our short term. So imagine, even though it doesn't say it up here, these are our, let me press the button here. These are our chemical buffer systems. Okay, how do these chemical buffer systems work? It's all right here in the first bullet point. Before I do that, let me just give you a definition of what a buffer system is. Just make sure I just know what I'm talking about. Okay, so I'm going to start with this slide because I think this will be a better way to start the discussion. Let's first talk about what a chemical buffer system is. And again, in the lab video for the buffer systems lab, I'll be reviewing this as well. And that's a good thing. So the more times we can review it, the better. So let's first do a definition. What is a chemical buffer system? A chemical buffer system is a group of chemicals that circulate in the bloodstream. That's what it physically is. We have three types of chemical buffer systems that we're going to talk about. And each of them is a group of chemicals. And this specialized group of chemicals circulates in the bloodstream. That's it. We have a group of chemicals, and they hang out together, and these groups of chemicals circulate in the bloodstream. Their only job, is to help buffer against pH changes. All right, so what's a chemical buffer system? It's a group of chemicals that circulate together in the bloodstream. All right, good, we got that. What does each type of chemical buffer system have? Each chemical buffer system has to have a weak acid, a weak base, and some hydrogen ions. So let's start there. Whether it's the phosphagen chemical buffer system, whether it's the carbonic acid bicarbonate buffer system, doesn't matter. Every chemical buffer system, of course, which is a group of chemicals that circulate in the bloodstream, every chemical buffer system has to have three components: a weak acid, a weak base, and hydrogen ions. Notice we need weak acids and bases, not strong ones. All right, now I'm going to go back. So we know that a chemical buffer system has a weak acid, a weak base, and some hydrogen ions. here's how a chemical buffer system works. And really, sometimes the body is amazing in its complexity. Other times we can talk about the body being amazing in its simplicity. So we know that pH balance is a very complicated thing because we have so many different things that can threaten to make our blood too acidic or too basic. We've got all these different places in our body that have different threats. But when it comes down to it, a chemical buffer system is actually beautiful in its simplicity. So here's what happens. How does a chemical buffer system work? A chemical buffer system is able to resist a large change in pH because it can act as either an acid or base, depending on what's needed to bring the pH back to normal. Let's give an example. A more common example, because there's more threats to lower acidity or to lower pH, if blood pH begins to drop, and that could be bad, what is the body going to do about it? If the blood pH, didn't even put pH because I'm so excited about what I'm going to say. If the blood pH begins to drop, what a chemical buffer system can do, so remember, we have these chemical buffer systems that contain these three components. So if the blood pH suddenly begins to drop, what the chemical buffer system does is it releases the weak base. So if the blood pH is becoming too acidic, I release the weak base. So I release the opposite. And what's a base going to do? A base is going to pick up hydrogen ions from the solution. That's going to fix the initial problem. The initial problem was that blood pH was too low, so we had too many hydrogen ions. So if I have a problem where there's too many hydrogen ions starting to build up, I can release the weak base, which will pick up hydrogen ions and temporarily counteract that potential drop in blood pH. Well, what would happen if the blood became too alkaline? If for a moment the blood became too alkaline, the chemical buffer system would release the weak acid to temporarily counteract the change. Wow, beautiful in its simplicity. I've thought about for years trying to think about some kind of real life example to try and bring this to life for people that had a hard time grasping it. And I have yet to come up with a really great real life example. The only thing that I've come up with, and I don't know if it helps anybody, I'm going to say it anyway. If you imagine you're cooking a sauce, you're making a soup or you're cooking a sauce, and we have parts of that sauce, like, how about a barbecue sauce? That's a good example. Maybe because we're all stuck at home now, we're making more food and you're making a barbecue sauce, right? And there's different components to that barbecue sauce. Maybe there's some components where you want to add salty flavors. And then there's components where you want to add sweet flavors, maybe a little sweet versus tangy. So we can imagine if you start to cook that sauce and it gets to be too salty. So let's just, for sake of argument, say that we could say that if the blood becomes too acidic, that could be a similar thing as if the sauce becomes too salty. In the short term, what can I do to fix that? If the sauce becomes too salty, I add something sweet to make it less salty. Just like if the blood becomes too acidic, I add a little base. Okay, I'm not sure if that helps or not, but maybe it does. If not, just completely ignore me. I have no problem with that. Okay. Last thing I want to say before we talk about specific examples is why we want each chemical buffer system to have weak acids and bases. So the idea is we have these three components, and depending on what's going on in the body, we may need to activate one versus the other. So these have to be weak acids and bases. So when we, in this example, I said if blood pH drops, we're going to temporarily release the weak base to pick up some hydrogen ions. We want these chemical reactions to be not complete. So that's why we have weak acids and bases. So in this example, the weak base will pick up some hydrogen from the surrounding solution, but not all. And that better makes these chemical buffers be able to use a weak acid or a weak base. Because in the body, the blood can become too acidic, and then half an hour later it could be too alkaline. So we need to be able to have these reactions where we temporarily release a weak acid or we temporarily release a weak base. We have to have them be reversible. And that's why it's helpful or needed, necessary to have weak acids or bases. If it was a strong acid or base, the reaction would be too great. We would be releasing, in an acid, we'd be releasing hydrogen completely. And that would make it very difficult to pick up that hydrogen again if we need to, a little bit later, add a base. So having a weak acid or base makes these reactions not complete. And that means that it's easier for us to be able to reverse them and be able to release a weak acid or base at a given time. All right, hope you're still with me here. We're going to talk about two examples. So there are actually three chemical buffer systems. We have the carbonic acid bicarbonate buffer system, we have the phosphate buffer system, and we have some proteins, some amino acids that can function as a buffer system. In this PowerPoint, I'm only going to talk specifically about this example. So we're going to just give you an example because I want to give you an example of what particular weak acids and bases are released and partially dissociating. So here we have the carbonic acid bicarbonate buffer system. What do you need to know? This is the chemical buffer system that is most active in our extracellular fluid. What we're going to find out is the phosphate buffer system. That's going to be a buffer system that we talk about in the lab portion of the buffer systems lab. And the phosphate is more active in the intracellular fluid. So we tend to have chemical buffers that are specialized to be able to act best in the extracellular versus the intracellular, because there's different environments and different considerations for each. Okay, so here we have the carbonic acid bicarbonate buffer system. This is an example of a chemical buffer system, a group of chemicals that circulate in the extracellular fluid. Like every other chemical buffer system, it has to have three components: weak acid, weak base, and hydrogen ions. I want you to look at the equation on the bottom. You are not going to be responsible for reproducing this whole equation letter by letter and number by number, which if you're like me, that's good news. But I do want to look at it for a visual effect. So what can we identify by visually looking at this equation for the carbonic acid bicarbonate buffer system? Well, we can identify that there is a weak acid. H2CO3 is carbonic acid, which is a weak acid. We can identify that there's a weak base. HCO3 is bicarbonate, or a weak base. And lastly, we can see that there are hydrogen ions. So this chemical equation shows us visually all three components that are necessary for a chemical buffer system. Weak acid, in this case carbonic acid, weak base, in this case bicarbonate, and some hydrogen ions. By the way, that's why it's called the carbonic acid bicarbonate buffer system, because those are the main components: carbonic acid, weak acid, bicarbonate, weak base. I also want to point out the arrows Because it shows us that these reactions are reversible. Because depending on a given situation, sometimes we want to purposely release the weak acid. Other times we want to purposely release the weak base. All right. I'm going to give an example. So let's talk about what happens in the carbonic acid bicarbonate buffer system if the blood becomes too acidic. Let me actually go ahead. I think the next slide is really what I want to talk about. Yeah. So, as I tend to get sometimes, here are two examples where I purposely got in the weeds. So this talks about what's happening in the kidneys. You don't need to know this specifically. This is what's happening in terms of CO2. You don't need to know that either. I just got a little carried away. If you're interested, this is some good stuff. But for our exam, you don't have to know it. Here's what I'm going to talk about. This is the exam stuff you should be reviewing. Okay, as always, I'm trying to be clear on what's most important. So what I'm talking about, what I'm marking with the pen and underlining is most important. Case in point, here's an example. So at the bottom, we have our formula for the carbonic acid bicarbonate buffer system. Here is carbonic acid. This is our weak acid. Here is bicarbonate, our weak base. And we have hydrogen ions. And we have arrows that are reversible. Great. So here's our example. What happens when blood pH drops? So if blood pH drops, it's becoming too acidic, what do we want to activate from this buffer system? The weak base. So remember, if the blood pH starts to drop, we want to activate the base to help temporarily counteract the acidity of the blood. So if the blood starts to become too acidic, we're going to release the base to counteract it. That's the first thing to know. I could ask you that question. If the blood pH begins to drop, what's going to be temporarily released in the carbonic acid bicarbonate buffer system? And you would say weak base or bicarbonate. What I also want to point out is when the blood becomes too acidic and when we temporarily release the weak base, this whole equation shifts to the left. So you should know the equation shifts to the left. I'm not going to ask you to write down the equation, but I could ask you which direction the equation shifts. So as we're looking at it, when the weak base temporarily dissociates, this equation is moving to the left. There's actually a sequential series of chemical reactions where the weak base dissociates and goes through a bunch of stuff. You don't have to know all those sequential steps. But theoretically, ideologically, when we look at this equation, when the weak base dissociates partially, it shifts to the left. And here's what I want to bring up what I brought up before. I said before, when a base dissociates, because what does a base do? A base picks up hydrogen from the solution. So when a base picks up hydrogen from the solution, one thing that it can do with it is form water. And that's what's happening as we visualize this equation. Our initial problem was that the blood pH started to drop. The blood becomes too acidic. To temporarily counteract that, we're going to release the weak base. What's happening chemically in a weak base is that the weak base picks up hydrogen from the solution. Wouldn't that be a good thing? When the blood pH drops, remember, when pH is low, we have a lot of hydrogen ions. So here's our initial problem. Blood pH drops, too much hydrogen. So when the weak base dissociates, it picks up hydrogen from the solution. So it's taking hydrogen out of the blood. And what does it do with the hydrogen that's taking out of the blood? It temporarily forms water. And this is a good thing because it takes up hydrogen from the solution and temporarily stores it as water, we've actually helped the situation. We initially had too much hydrogen in the blood. I released a base which actually took hydrogen out of the solution of the blood. All right, cool. Keep it simple. So, okay. I didn't even put a slide here because if you memorize it one way, I think you can automatically know the other way. What if the example was in reverse? What if, let me just do a little sidebar. If blood pH starts to rise abnormally, what are we going to dissociate? Or what are we going to release? We're going to release the weak acid. And what direction is this equation at the bottom going to move? Equation moves to the right. So if our initial problem, to the right, if our initial problem was the pH of the blood becoming too alkaline, we would temporarily release the weak acid. When we move the equation to the right and we release the weak acid, we're adding hydrogen to the solution. Our initial problem was the pH was too high. When pH is high, we don't have enough hydrogen. So by the weak acid dissociating and the equation moving to the right, we release weak acid, which temporarily brings the pH back down to normal. So you should know if the pH goes up and down, whether the weak base or weak acid is released, and then you should know whether the equation moves to the right or to the left. If you have questions, reach out to me, please. Okay. Let's see, because I want to talk. Okay. Sorry about, hopefully you're not getting motion sickness because I'm moving too quickly through here. All right, so I told you that there were 3 chemical buffer systems in the body. The first chemical buffer system is the carbonic acid bicarbonate buffer system, and that is more active in the extracellular fluid. A second chemical buffer system is the phosphate buffer system. This is more active in the intracellular fluid and the urine. It also contains a weak acid and a weak base and hydrogen ions. It also moves to the right or left. I will not be asking you specific questions about this on the exam in terms of the equation, but you should just know that the phosphate buffer system is a chemical buffer system, and it acts more so in the intracellular fluid. When you go through the lab this week, the buffer systems lab is utilizing the phosphate buffer system. Lastly, we have a protein buffer system. It's a little bit different in how it does it because instead of having molecules of weak acids or weak bases, the body's able to utilize certain amino acids because it turns out certain amino acids are able to accept or release hydrogen. And because they have the ability to do that, that makes them very well positioned to be buffers. I'm not going to ask you any questions about this on our next lecture exam. Okay. Here's what I want to talk about next. So we talked about our 2 types of chemical buffer, our 2 types of buffer systems. Our first buffer system was a chemical buffer system, and this was short term. So we utilize these chemical buffers in the short term. But if we only had that, we would never be permanently getting rid of excess hydrogen, for an example. So if the blood becomes too acidic, because you've got a buildup of ketones or lactate, the chemical buffer systems in the short term can help buffer against the whole blood becoming acidic. But that short-term chemical buffer system is not permanently getting rid of anything. It's a Band-Aid. So we're always going to be shifting from a chemical buffer system, which is useful in the short term, until we can activate our physiological buffer systems, which is what is more permanent and is our long-term solution. So we're always going from chemical to physiological, because that's how we're going to get rid of things permanently. And as a matter of fact, you already know our physiological buffer system. So this is going to be a pretty quick discussion. What two body systems that we've discussed this semester play a major role in pH regulation? Ding, ding, ding. Respiratory system and urinary systems. So the chemical buffer systems are down in the trenches in the blood in the short term, helping to buffer against any pH changes out of that range. But we're always going to be relying on our respiratory system and our urinary systems in order to permanently get rid of hydrogen or CO2 to better regulate against pH changes. So exciting. We know how these work, and these are much more powerful for the record. The chemical buffer system is not very powerful. Its benefit is only that it is short term and it is immediate. The physiological buffers are so much more powerful. So if I were to ask you a question, which type of buffer system is more powerful in regulating against pH changes, chemical buffers or physiological buffers? The answer would without a doubt be the physiological buffers, much more powerful. So we already know how the respiratory system can help to alter pH by releasing more or less carbon dioxide. So if for any reason I've got a buildup of hydrogen or CO2 in the body and the pH is starting to drop, we're going to want to increase respiratory rate to get rid of more CO2. But of course that takes time. It takes time to send a message to the brain, to then send a message back to the muscles for breathing to increase respiratory rate. So we utilize the chemical buffers in the short term until we can get the messages to increase respiratory rate. And then how does our urinary system work? Well, we can regulate pH by excreting more or less hydrogen into the urine. If this example down below, if the blood pH is increased, hold on, I don't like how that's written. Let's give you a better example. Because it's more, if blood pH drops, if the blood pH becomes too acidic, we're going to want to release more hydrogen in urine. Right, so we have blood pH that drops, and of course when the blood pH drops, we have too much hydrogen, because they go hand in hand. So one way to fix that, having too much hydrogen, is to release more hydrogen in the urine. Even compared to the respiratory system, our renal buffer, our urinary system buffer, is the most powerful of them all. And we know why. You can see it at home here. I'm widely gesticulating. My hands are all up in the air because it's so exciting and it's so true. We already know why the renal system is the most powerful, because the kidneys filter 150 to 180 liters of blood. So because it has so much blood coming through them, the kidneys, they can have an ability to release a whole lot of hydrogen and regulate a whole lot of pH. So exciting. Okay, we're coming down the home stretch here. Just like we talked about electrolyte imbalances, if we have hyponatremia or hyperkalemia, of course we can also have acid and base imbalances. So we're going to be pretty general with this. We know that probably acidosis is a condition when the blood pH drops below our normal range. On the other hand, we know that alkalosis is when the blood pH goes above the normal range. Typically, our body is pretty good at maintaining blood pH, so it does take a lot to get into an acidotic situation or an alkalotic situation. But of course, it can most definitely happen. So what I want to do is I'm going to go through an example. This is not going to be on the test. But because we spent so much time talking about electrolytes in the cells, I think it's really worthwhile to just go through what could happen if the blood becomes too acidic in terms of ions. Okay, so I'm going to draw a neuron. And again, this is not going to be on the test, but I think it's a useful exercise in seeing what the long-term or what the consequences are of having acidosis. Okay, so we know this is a neuron. So inside here would be the intracellular fluid, and then the blood outside of it would be the extracellular fluid. So what happens if we have acidosis? Acidosis means that the blood pH drops. And when the blood pH drops, we have too much hydrogen in the extracellular fluid. While we expect some hydrogen in the extracellular fluid, in an acidosis condition, when the blood pH is dropping, extracellular fluid, blood pH, we have too much hydrogen. So what's going to happen? Well, what's going to happen is the cells are going to try to counteract this and make the best of it. Let's see, what color can I use? Let's use green. So we know that there's more potassium inside the cells. So one way that the cells are going to respond when there's all of a sudden way too much hydrogen in the extracellular fluid, to maintain homeostasis, because we've got too many positive ions on the outside of the cell, we're going to start to diffuse potassium from the inside of the cell to the outside. Actually, let me go back because I missed a step here. It's pretty important. Okay. As a result of having too much hydrogen outside of the cells, we now have a larger concentration gradient of hydrogen. So now we're going to have hydrogen all of a sudden moving from high to low concentration. And that could be bad. That's going to disrupt our cell membrane resting potential. We know that the inside of a cell has a negative charge relative to the outside. That potential energy is what enables our cells to generate a nerve impulse, which is very important. So if I have an accumulation of positive ions on the outside of the cell, increases the concentration gradient, hydrogen is going to start to flood into the cell. And that could be bad. So now we know what's going to happen because I already gave you a preview. To counteract all these positive ions flowing into the cell, potassium is going to start to flow out of the cell. But what happens if all of a sudden a bunch of potassium flows out of the cell? We make the inside of the cell more negative than usual. If you want to, and you may not want to, but for those who are daring, if you want to step back into time, into the resting membrane potential from AMP1 or from the discussion of nerve impulses, we know that potassium flows out really fast. So potassium flows out faster than other ions. So we're going to lose a whole bunch of potassium. Normally, the inside of our cell is -70 or -90. If I lose a whole bunch of potassium, now, instead of the inside resting charge being -90, now it's -110. And remember, and this is a really bad neuron, by the way. In order to depolarize the membrane to make an action potential, we had to turn the inside from -90 to +30. But what if now I'm starting at -110? Now the neuron's got to work harder. It's got a longer distance to go to get to positive 30. And this is why when we have acidosis, because of the flow of hydrogen ions into the cell and the outflow of potassium out of the cell to try and make up for it, which lowers the resting charge, it makes it harder to make a nerve impulse and make a muscle contraction. So acidosis is going to lead to problems where we are unable to generate enough nerve impulses or muscle contractions. If you were to play this out in the opposite, with alkalosis, the inside resting membrane potential would actually be higher, and it would make the cells too easy to make a nerve impulse or muscle contraction. So that's why it says here, makes the cells hyperexcitable. I am not going to ask you that detail. I just wanted to lay out for you what can happen once we have an accumulation of hydrogen ions. It can influence a whole bunch of other things like a domino effect. What I do want to talk about, and we're almost getting to the end here, everyone. I know. Okay, what I do want to talk about is the two primary causes of an acid or a base imbalance. So we talked about alkalosis and acidosis. It turns out that there's typically two causes of pretty much any kind of alkalosis or acidosis. One of the causes is respiratory. Respiratory problems can very easily lead us into an acidotic condition or alkalosis to a lesser extent. And the other common cause is metabolic. So it's pretty cool. And like I said before, the body can be so complex, but sometimes it's capable of just being beautiful in its simplicity. I'm not saying that alkalosis and acidosis is beautiful. But when we have alkalosis or acidosis, it's typically from one of two causes, a respiratory cause or a metabolic cause. And I'm going to give you two examples that show you this. Let me go back, because the most common way that we have acidosis is because of a respiratory acidosis. This is the most common problem, respiratory acidosis. Think of COPD, emphysema, congestive heart failure. Think of in a short-term asthma attack. The body is not able to get rid of enough CO2. And because we can't get rid of enough CO2, carbon dioxide accumulates in the body, which lowers the pH. And boom, we have respiratory acidosis. But the reason why it's called a respiratory acidosis is because the cause was of respiratory nature. There was some kind of problem with our respiration, particularly breathing out, where we can't get enough CO2 out, so it accumulates and it turns the blood acidic. That's a respiratory cause. The second most common acid or base imbalance is metabolic acidosis. Second most common. And this is when the blood becomes acidic because of the accumulation of some type of metabolic byproduct. Two common examples would be lactate and ketones. So here the cause isn't respiratory. It's not a cause of the blood becoming acidic because of CO2. Here the blood becomes acidic because of a buildup of certain metabolic byproducts, typically ketones or lactate. So the blood becomes acidic because we have an accumulation that we're not able to get rid of some kind of chemical reaction byproduct. The next couple slides give you how the body compensates for it. We've already talked about this, so this is not something you need to review in this slide in particular. This is a little graph. You don't need to know it for the exam. What I want to point out is, again, you're not going to be on the exam, but what I want to point out is, just FYI, if someone has a metabolic acidosis, we have a buildup of lactate, which turn the blood acidic, Usually the compensation in order to fix that problem is the opposite. I mean, if our initial problem was because of a metabolic problem, we're not going to be able to use that same problematic metabolic thing to fix it. So we tend to use the opposite. And the same is true for respiratory. If we have a respiratory acidosis, we're going to tend to utilize our metabolic processes to fix it. But this is not going to be on the test. Okay, last thing of the presentation here. Not going to be on the test. If you want to just shut the video down, feel free. I'm not going to be offended. I have students who don't listen to me all the time. I'm used to it. I only bring this up because, not that you really care about me so much, but I had a situation years ago. It was my first marathon? No, my second marathon. It was up in Vermont City. It was up in the Vermont City Marathon in Burlington, Vermont. And I actually passed out towards the finish line. And here is the actual marathon medical record report. So it took me right to the medical tent. And as a case study, I think it can be pretty interesting. Pardon me. But if we think about what was happening, this race took place over Memorial Day weekend. So I had trained all winter and spring, and it was relatively cold out. And then that year, was it 2008, maybe 2008? That year, it was abnormally hot. Even in Burlington in May, it was like 85 degrees. So despite me being well trained, I was not acclimated to the temperature. So even though I hydrated well, I was sweating at a greater rate than I was used to. And at about mile 25, I stopped sweating. I started to lose consciousness a little bit, and I couldn't really tell where I was running. I don't remember crossing the finish line. They took me right to the medical tent. But there's some interesting information here. So again, not going to be on the test, but just kind of a fun case study if you're interested. Why would I have had, look at they circled altered mental status. Why would I have had central nervous system problems from being dehydrated? Because I had an electrolyte imbalance. So I was slightly hyponatremic. And as we talked about before, we know that sodium and potassium their movement is what's required for a nerve impulse conduction. So once you start messing with the normal amounts of sodium, it messes with our ability to generate a nerve impulse. They took my blood, and they did right there in the medical tent, blood sodium, blood potassium. I was slightly hyponatremic. I think below 135, I want to say, is hyponatremic. So I'm going to say slightly, because you're probably going to see a lot of numbers that are worse than that. I was definitely hypokalemic. Normal blood potassium is about 5, and I was at 7.8. So I was definitely hyperkalemic. And it was because of these electrolyte disturbances that I was having nervous system symptoms. They did, let's see, so this is 1 o'clock p.m. and 1:22 p.m. I just ran 26.2 miles, yet my blood pressure was low. Why? Because I was dehydrated. So my body just didn't have as much fluid to be able to pump out. When blood pressure drops like that, the pulse usually goes through the roof, which it was really high after I finished. So there wasn't as much blood to pump out, so the heart was trying to pump out harder to make up the difference. Pretty high respiratory rate. I definitely had a high body temperature and a slightly lower oxygen saturation. Okay, so just a little extra if you're interested. That's all we have for this one. Good luck studying. If you have any questions, do not hesitate to reach out.